Good afternoon all. This video will give a brief overview of my poster on assessing the impact of variants in giant sarcomeric proteins with computational methods. So first of all, why should we be interested in this topic? Well, diseases of the muscles are responsible for significant damage both to individual health and to healthcare systems, and better strategies are needed to address them. Importantly, many of these conditions have a heritable component, which have been linked with the proteins that make up the sarcomere, the vital structure in which the muscle cells that allow for contraction. So, being able to predict the structural and functional consequences of a given mutation in these proteins would improve our ability to determine their capacity to cause disease, or pathogenicity. However, it is notoriously hard to determine which variants will be damaging in these proteins for several reasons. First, myopathies are complex conditions affected by other non-genetic factors such as diet, age, and so on. Moreover, these proteins are extremely large. The full isoform of titin, for example, is over 300 domains in length, or 36,000 amino acids. This means that not only is it impossible to experimentally characterize all possible consequences of mutation, it also means that healthy individuals will also carry rare variants, complicating variant prioritization. As a result, there is limited experimental validation of variants prioritized by high-throughput studies, such as genome-wide association or whole exome sequencing. However, this also means that this is an area in which computational approaches can have a considerable impact. So, we started with the goal of computationally assessing variants in the giant protein titin, currently the most well-characterized, but still largely unexplored of the sarcomeric proteins. There were two main questions that we sought to better explore through this work. The first was to determine what insights could be gleaned from the most recent and reliable predictors of pathogenicity when applied to a large and mostly uncharacterized protein such as titin. For this purpose, we use the pathogenicity prediction algorithm Rhapsody, a predictor that takes into account many different levels of information to increase the accuracy of its predictions. We used a template-based homology modeling pipeline to generate models for each of the 301 immunoglobulin and fibronectin domains in titin. Then we predicted all possible single amino acid substitutions at every coding position in the protein, or roughly 550,000 individual predictions, as an in silico saturation mutagenesis approach. These were then arranged by domain to give us a larger picture of how the patterns of predictions compared between and within domains. The second question was to determine how we could improve the accuracy of our predictions further, using information and strategies not used by previous predictors. We wanted to use existing knowledge about the domains in these proteins and transfer this information and understanding to the very similar but less well-characterized domains of the same or similar folds. We also added additional features to the classifier based on all-atom molecular dynamics simulations to see if these dynamical features could increase the discriminative ability of the predictor. So, after compiling the pathogenicity scores computed by Rhapsody, we visualized the predictions as heat maps, where more likely damaging variants were displayed as red, and least likely damaging were displayed as green. You can see if you zoom in on these plots that the pattern of most damaging variants tends to match well to patterns of solvent accessibility in purple and secondary structure elements here in blue. This is to be expected, as more buried residues would naturally have more of an impact on protein structure if altered. We can also see from the count charts that single positions tend to have either most or very few of their possible substitutions predicted deleterious. This tallies with the idea of structural hotspots of pathogenicity. Additionally, we noticed a curious trend that patterns of prediction in a specific region of the I band, immunoglobulin domains 31 to 82, show a different pattern of hotspots to other regions. As can be seen in the charts, there is visually more green and thus more non-damaging variants predicted in this region compared to immunoglobulin domains in other regions. This is surprising as all of these domains do share a very conserved topology, and predictors that rely only on structural information, such as MCSM, do not pick up on this discrepancy. This region is, as can be seen here, a phylogenetic clade within titin domains, and corresponds to a region thought to be responsible for some of the elastic properties of titin, involving unfolding and refolding of domains. We propose that this pattern of predictions reflects a greater flexibility in the structure and topology of these domains, and plan to investigate this question further. For the classifier, we calculated sequence-based features used by most standard variant classifiers such as SIFT and PolyFen2, and added them to the structure and network-based features that are computed by Rhapsody, as well as our additional dynamics-based features. After training the classifier, we compared the performance of popular predictors, Rhapsody and Condell, on a test set of known pathogenic missense variants. While all three predictors performed well and our predictor showed only a slight increase in performance over the other two, we were able to correctly discriminate some cases that proved harder for these other predictors to negotiate, such as the leucine to proline variant in fibronectin 20. You can see here in this principal component plot that the trajectories of the two move to occupy different conformational space. Thus, we have established additional examples of systems where dynamical features can add discriminative ability, and that further progress with this sarcomer-specific classification is worth pursuing in other sarcomeric proteins and systems, and that more data and experimental validation of examples should be prioritized. Thank you very much for listening. If you have any questions, please do send me an email or a message, and do enjoy the rest of the conference. Thank you.